So now I want to take just a little bit of time to review gene expression, the idea of a gene, and go back and talk about the differences, major differences, overview between prokaryotic life and eukaryotic life when it comes to gene expression. Now, we have to go back to our domains. We talked about very briefly when we started talking about cells. Gene expression is a fundamental process. That's why it's called dogma. Right? It's a dogma of molecular biology. All life has information in DNA that is transcribed to RNA, that is translated into a protein, and proteins do things. Now, Archaea are a group of prokaryotes. So remember we talked about the three domains of life, the three biggest groupings of life are bacteria, archaea, and eukarya. And most of the things that you know and love are eukaryotes, multicellular, your dog, your cat, your tree, your grass, your brother. All these are eukaryotes. What we think about as bacteria actually fall into two categories, prokaryotic life, bacteria, and archaea. Now, again, the fact that these are separated on a domain level suggests that there's something very fundamentally different about archaea and bacteria. And ultimately, the difference lies in how closely they follow a eukaryotic model for gene expression. So what we're ultimately going to see is that archaea are a little bit more like eukarya in many aspects than they are like bacteria, and that merits then, one, a different domain, but two, also, if you see this phylogenetic tree from the earliest organisms, a closer relationship to eukarya than eukarya are to bacteria. So bacteria and eukarya differ in their RNA polymerases. The polymerases are different. Termination transcription is a little different. The ribosomes are different. This is why, though ribosomes are the universal site of protein synthesis, there are slight differences. This is why antibiotics work, right? Because many antibiotics work by a mechanism to shut down prokaryotic ribosomes. And so if I flood your body with a chemical that stops protein synthesis, it better work on just prokaryotes, bacteria, and not on you. And what they do is target the differences between your ribosomes and prokaryotic ribosomes. Otherwise, it would just be poison and it would kill you. So archaea tend to resemble eukarya more in these respects. So now at the same time, bacteria can simultaneously transcribe and translate the same gene. So in eukaryotes, we have the nucleus and then the cytoplasm. So archaea, being a prokaryotic cell, are more like bacteria in this respect. So again, a picture of this, I had alluded to this earlier, this idea that you can literally have RNA polymerase working on the DNA, making a copy into RNA, and then the ribosomes binding to that before it's even done. And what this leads to is an efficiency of speed in producing proteins. And when you're a single cell with no compartments, that is your greatest advantage, numbers. Can I make more of me? Can we overwhelm competition? And that's what bacteria are very, very good at, reproducing very quickly. So we go back to this idea of a gene as a universal concept, and we kind of see that a gene is more than just DNA information that encodes a protein, right? So we can call it a discrete unit of inheritance using a non-molecular definition. We can go and say it's a specific nucleotide sequence in the chromosome, but that nucleotide sequence having a promoter and a terminator, and what we're going to see later, enhancers and other elements that turn the gene on or off, it's more than just the nucleotide sequence for a polypeptide chain, though we'll often use the term gene to refer to just that sequence as well. So when you go on in molecular biology, and we'll talk about this a little bit, but it's a huge field, this burgeoning field, the idea that there are many, many genes with products that aren't proteins. In fact, the RNA that is produced is in fact a protein. And so what we're really going to define then is a gene as a region of DNA that can be expressed, quote unquote, to produce a final functional product. And that product ultimately is going to be a polypeptide or an RNA molecule. We certainly are going to focus on the idea of proteins for now in our intro understanding of gene expression.